So during pregnancy, the smooth muscle layer of the uterus, or the myometrium, undergoes a program of differentiation and adaptation at the molecular and cellular level. In parallel with this myometrial programming, the myometrium and the fetal membranes also undergo an immune transformation, specifically marked by the activation of the nuclear factor kappa B signaling pathway. The consequence of both of these processing occurring in tandem is the production of a tissue that can generate coordinated and powerful contractions at labor. This process must occur in a timely manner to ensure proper organ development of the fetus at term is completed for survival outside the uterus. Unfortunately, there is still a lot that we don't fully understand about what is underlying these processes to coordinate myometrial programming and immune transformation during pregnancy. What we do know is that during pregnancy, the immune system goes from a pro-inflammatory state at implantation into an anti-inflammatory state throughout pregnancy, and then back into a pro-inflammatory state. So what I did was I took this pro-inflammatory state at labor and I simulated it using interleukin-1 beta, and I used interleukin-1 beta, incubated them with human myometrial cells to look at the NF-kappa B signaling pathway and heat shock protein expression within these cells. Small heat shock proteins are a family of 10 proteins are involved in regulating stress responses. HSPB5 and HSPB1 are known to regulate immune transformation and contractile activation in various tissues. In particular, HSPB5 may activate NF-κB, but the mechanism of action of HSPB1 and HSPB5 within the myometrium remains unclear. As well, extracellular vesicles are emerging as an important means of intracellular communication in tissues capable of transporting cargo, including HSPs from cell to cell, and they could play a role in myometrial activation. So when I stimulated the myometrial cells with interleukin-1 beta, it was shown that heat shock protein B5 and B1 shot up right away, so increased expression right at half an hour. This correlated with a downregulation of IKB, which is the inhibitor of NF-kappa B. NF-kappa B in this diagram that you see is the red P65-CrEL P50 complex. So when this inhibitor of kappa B, IKB, moves away from this complex, it is allowed to enter the nucleus of the cell to start gene transcription for various proteins involved in inflammatory activation in labor. Furthermore, these inflammatory activators were shown to be packaged up in cargo and extracellular vesicles for the first time ever that extracellular vesicles were released from human myometrial cells. So these results demonstrate a novel mechanism of the role heat shock proteins and NF-κB signaling pathway play in the initiation of labor within the myometrium, and how EVs work to aid in cell-to-cell communication to generate a powerfully coordinated contractile tissue at term.